So I'm going to talk about degraded DNA and what a representative trial testimony looks like. And then I'd like to leave lots of time for questions and answers. Okay. So this was the second case that Cybergenetics was involved in with TrueAllele in Pennsylvania. Reading is in the eastern part of the state. And there was a man, Glenn Lyons, who was accused of killing a woman on the grounds of a hospital and was held for court. Just to sum up the case as was described in the newspaper at the time, Lyons was accused of killing Kathy Liebig, whom police said he was having an affair. Liebig was found in the back of her SUV in a parking lot near the hospital. She had been stabbed 30 times, according to investigators. So I'll tell you how the evidence was found once we get there. And this is a sweatshirt. Mr. Lyons was a parolee, and he had a relationship with the victim, and it was believed that as she was trying to explain why she was going to break up with him, he attacked her. He was also a drug addict, and after she was killed, he disappeared on a 3-day drug spree in the eastern part of the state. And along the way, he was seen on closed-circuit TV dropping off a bag, and that bag was then later found at a garbage dump. It was actually found by a team of police cadets who were recruited from the academy for this. The garbage dump shut down for a few days, and as they were nearing sundown of the last day, where the operator of the garbage dump said he would let them keep the place closed, they found a plastic garbage bag that contained some of the victim's personal effects, along with this sweatshirt. The sweatshirt had blood spatter on it in patterns that indicated that an assault had taken place. And in the neck area, the back of the neck, there was a DNA mixture that was ultimately shown to contain 75% of the victim and 25% of some other person. So I want to go through what degraded DNA is, just so that we're all talking about the same thing. Here's a DNA molecule. Think of it as one of those paragraphs that Dickens was writing about years ago, when he was writing about DNA. And from this one locus, that molecule can be amplified by reading from the beginning of the paragraph and the end of the paragraph. And the way polymerase chain reaction works, which is the heart of STRs, is that it reads from both sides, it makes copies, and then it keeps doubling the number of copies. So every time it makes a copy, the copy can be copied. So you get 2, 4, 8, 16, 32 copies, and the DNA is labeled, and that's how you can take a trace amount of DNA and end up with millions of copies that can be detected by machines. And a normal DNA signal has loci all the way across in different size windows. The original DNA kits looked at one locus, and then they looked at multiple loci where they arranged the paragraphs to be of sizes that didn't overlap with each other. That's how you get multiple readouts. And then they went to multiple dyes, and that's why you can get 10 or 17 loci from one tube. You're using different size regions and different colors, blue, green, orange, different fluorescent dyes. And what you see with normal DNA signal is that across these different loci, the signal is pretty even, because you're getting roughly the same amount of DNA being amplified from the text, even though the paragraphs are increasing length as you move from left to right. What happens with degraded DNA is there's a cut. And that is usually caused by enzymes from bacteria. It can be due to other causes, radiation, and chemicals. And once there's a cut, the copying mechanism doesn't work anymore. And so PCR can't read through a break and that's degraded DNA. There's a break in the molecule. The molecule can't read through it, and so even though the DNA is present, once there's a break, it's invisible to the detection process. It's not seen anymore, even though that amount of DNA is present. And this is the key concept of what degraded DNA looks like as a signal. The chance of a break is pretty even on a per sentence basis. So for every 100 letters, it's the same frequency of there being a break. If you have a very short sentence, then because the chance is proportional to the length, the longer the sentence is, the more opportunities there are for a break. Okay, so a short fragment, a short sentence is more reliably amplified and detected than a longer one, because a longer sentence has a greater chance of at least one break. You can have multiple breaks, but as soon as there's one break, it's not detected. And so what happens is as the molecules get longer, as you go from left to right, the chance of actually observing degraded DNA goes down with the increasing size of the molecule. And the pattern that's seen in the data is a fall-off where the peaks are no longer of the same height. You're seeing that at the longer sentences, the longer paragraphs, there's no signal. And that's because it's attenuated by having a greater chance of there being a break. So that's what degraded DNA looks like. And different contributors to a mixture can have different amounts of degradation. Some DNA, one contributor may contribute more intact DNA, and another contributor may contribute more degraded DNA, because they come from different people and at different times. TrueAllele solves for the different degradation of different contributors. This is the victim's DNA, which was not terribly degraded, but the other DNA that was left had more of a decay curve to it across all this data as we go from sentences of length 100 to 500 base pair. And the reason why degraded DNA is important for computers and people when you're interpreting DNA evidence is that when you look at the data, the proportions seem to change. Right? At the very beginning, you may have what is in reality a 3-to-1 proportion. But as there's less and less of one of the contributors, you may be observing something more like a 10-to-1 proportion. And if you're using mixture weight and the difference in quantities to help solve the genotyping problem, it's good for the computer to be able to use that information and know that even though it looks like it's maybe 9-to-1 instead of 3-to-1, that's because it really is 3-to-1, but there's fall off on one of the components. So that helps people as well when they analyze the data. The computer can do it more precisely. And that way you can get a little more information from the data than if you didn't account for that. Okay, in this case, the reason we were asked to be involved is the human statistic on the data looking at maybe 2/3 of the genetic loci and saying that the other third were not interpretable was 42,000, which is a 1 followed by 4 zeros roughly. And the computer's match statistic was nearly, a lot of zeros, about 10 trillion. So there's a difference in the number of zeros of roughly 9 zeros, about a billion-fold difference. And so the prosecutor in the case thought it would be helpful to have the computer interpretation, which had been used in a trial the year before in Pennsylvania. So what I'm going to show you now, you've seen a lot of slides that explain what DNA evidence looks like, and they've come out of the courses and trials that we've done. And so what you're seeing, this is a representative set of slides of what is the jury sees at trial. This is of historical value. It was the first time we brought together the materials from our courses and shown this. It's been a little refined over time, but the basic concepts and pictures are the same. And this was what was shown to the jury at Commonwealth versus Glenn Lyons in Reading, Pennsylvania, 3 years ago. We begin with the first slide, and this looks familiar, but I'm just going to go through, if you don't mind, the way we would explain this to a jury. A barrister is asking questions. Some of you may be so familiar with this after an hour or two that you could serve that role, but I'll just continue. So how is DNA evidence interpreted? Well, we begin with an evidence item, which is a biological sample, and then the laboratory takes that biological evidence and they extract the DNA. They put it through machinery that can amplify the DNA and make millions of copies of what's there, and then it's read out on a DNA sequencing machine or a genetic analyzer that gives signals where with increasing length we can determine alleles of what's present, and the heights give a sense of the quantity of DNA. And that's the starting point of the evidence data for any type of review. Whether it's done by a person or a computer, that's the starting data. And the underlying principles are the same. From the data, genotypes are inferred. Now, I'm going to focus on what computers do. As you've seen, people do it a little bit differently, but ultimately they are inferring genotypes. They're just less informative genotypes. What the computer does is that for every locus, for each contributor, there are a list of, by the way, I left out all the first stuff about 5 minutes of what is DNA, what is a genotype, what is a cell. That was done at this trial as well, but that was just answering questions. So the jury did know what DNA was, but we didn't have those slides back at that time. And so out of the 100 or so pairs of alleles, the data concentrates the possibilities down to just a few and provides probabilities that go along with those possibilities at each locus for each of the contributors. And that's an objective process that then comes to a full stop, is written down, and then can be used to compare with other individuals. And compare, in this case, say with a known genotype from someone, in whether it's the victim or the suspect, and make a comparison to see to what extent the known reference genotype, say a 1012, matches someone on the list, and with what probability, and with what strength of match. Okay, so that's the starting point of what the process is. And computers can use all the data. And here we're showing quantitative peak heights at locus D8, which means that it's on chromosome 8 in some region. It's a slightly larger, sort of mid-sized. The sentence ranges from 200 to 250 DNA letters long. And the size from left to right is giving the size of the paragraph. And the numbers on top, we have since made the numbers larger so people can see them, so I'll tell you what they are. This is a 13, a 14, and a 15. They represent the number of repeated four-letter words that are in the middle of that sentence. And so each allele is progressively longer because the number of repeats is longer, and those are the alleles that we have, 14, 15, and 16. The y-axis, which shows the quantity of DNA on a scale of fluorescence, indicates that some alleles are present as taller peaks in much greater quantity than others. In this case, alleles 14 and 15 belong to the victim, while allele 13 belongs to some other person. Okay, so that's the data. And in this case, since there was a human review done, it was important to explain how what the computer does differs from what a person might do. Okay, so people may use less of the data. They apply a threshold, and over the threshold, the peaks become allele events that are present, a 13, 14, and a 15. And under the threshold, anything else disappears. And sometimes the threshold does include the alleles from the genotype you want to compare against. and sometimes it doesn't. When it doesn't, what human review typically will do, at least in the US, is to say, let's not use that locus because it's not contributing to a match statistic. Okay, and under the threshold, we don't see alleles. And what the computer does is it tries out every possible genotype. When you have known references, and this can be more important when there are family members, for example, if you have sexual assaults of a parent against a child, or if you have a crime scene where there are 2 or 3 or 4 related people, parents and children that have all been killed, the computer can assume the genotypes of known victims. And actually, there's a procedure that we go through where you start off perhaps assuming 4 contributors, and then you find a very strong likelihood ratio, a statistical match to one of the people. And then you say, fine, so we know that person contributed their DNA with high probability. We now assume that that person is present and solve for the other 3. And we repeat, we do it again. We find another person who might match with a very high match statistic. And then we continue and say, assume 2 known people, the first 2 that we matched, and solve for the other 2. And in that way, we can peel away the layers of the mixture and identify, as we've done in some cases, the 3 or 4 people with appropriate match statistics who have contributed to the evidence. That's useful for investigative purposes, not just for prosecutorial purposes. It's just to know it. In fact, it's useful for defense purposes, like what's really going on and who contributed their DNA to the evidence. In this case, in gray, we had the computer assume that the victim was present. We first didn't assume the victim was present. I'll show at the end and establish that she was there. Once we know that she's there with a high match statistic, we can assume with these gray bars that the victim's present. That becomes an easier problem for the computer, a very easy one actually, to just solve for the one missing genotype. It's almost as easy as single source in many cases for the computer because it's locked in one of the genotypes. It's only looking for one other one. And so it tries out lots of possibilities. In this case, those little blue bars would move all over the place. They become big blue bars, small blue bars. That's the allele pair of the unknown individual who's the second contributor to the mixture, the minor 25% contributor. And this is just showing that the computer is doing more with the pattern than just the allele pairs. For those who are familiar with these terms, it's accounting for stutter, relative amplification, and other artifacts of the amplification process. And again, a better explanation gives a higher probability. And in the end, this is the evidence genotype. The victim is a very definite 1415, corresponding to those 2 very tall peaks. And then there's another genotype at this genetic locus where the probability has been concentrated onto 3 possibilities. And those are 13, 13, 13, 14, and 13, 15. As we saw here, if you have a 13 and a 14, then the low pattern of low, very high, and then high but not as high is explained reasonably well, which is why in explaining the data, that possibility out of 100, the 13, 14, ends up with most of the probability. Again, the system has not looked at the victim yet, or the suspect. This is just from looking at the data. Now a match comparison can be made. And this is how to explain a likelihood ratio to a jury in visual terms. Before we ever looked at the data, before the computer looked at the data, there were 100 of these brown bars that reflected the probability of a random black individual in Pennsylvania. And that was the random genotype. Blue is showing the evidence genotype after the data and computer thinking has focused probability. And now, for the first time, we take a look at Mr. Lyons' genotype, which happens to be the 1314, and it's outlined here in red. And we could have seen a decrease in probability from brown to blue, but in this case, we happen to see, without any bias, that there was an increase in probability from brown up to blue. from the random genotype up to the evidence genotype at the suspect's genotype. That's what the match statistic is. And that's a gain of roughly 6. And that's giving us the probability of an evidence match divided by a coincidental match. Those numbers are multiplied together across 15 genetic loci. And then we can answer the question, is the suspect in the evidence? And one can say that a match between the suspect and the evidence is 9.5 trillion times more probable than coincidence. In this case, the barrister wanted a statement about reasonable degree of scientific certainty, which they sometimes like in Pennsylvania. And we can agree that with a reasonable degree of scientific certainty that the evidence item Q9 contains DNA from the genotype of the suspect. Similarly with the victim, this is looking at as a 2 unknown genotype problem, that a match between the victim and the evidence is a quintillion times more probable than coincidence. She's in there. I mean, in plain English, right? It's a very strong match statistic. She's the major contributor, and so on. Now that 42,000 match statistic, the way RMNE works, applies to both the victim and the accused. It's like sort of a one-size-fits-all based on the data, as opposed to separating out I guess, 18 zeros, saying, well, the victim's definitely in there, and the defendant is in there with 13 zeros worth of match. In the end, he was convicted. He appealed to the Pennsylvania Superior Court, which is the appellate court, and then in the fall, the Pennsylvania Supreme Court, which is the court of last resort for appeals, upheld his conviction and death sentence. People rarely get executed in Pennsylvania. The appeals process goes on for the rest of their lives. Some states it's different, like in Texas. They actually execute a lot of people there. And his view on it is that he was framed, according to the circumstances and much other evidence not. A question that some of you may be asking is, how can I get started? And we have on our website, www.cybergen.com, we indicate the sort of data we need. We basically need electronic data files and some case context to know who's who and how to process a case. And I think we have about an hour. Now, I'd like to entertain questions.